how to proceed so we don't have to deal with interruptions. So we're going to do the whole thing. Now, I don't remember if I mentioned that last week. If you have a question that is super related to the material, feel free to ask. If it's a bit of a tangential question, I recommend write it down. I think I mentioned that if I pause the video, the recording is going to be screwed up. We don't want that, right? So if you have sort of write it down. I will be more than happy to answer your question or not, depending on whether I know the answer or not after the class, okay? But if you want me to slow down, if you want me to repeat something, feel free to yell and I will do that happily, okay? So, on that cheery note, let's talk about the whole process of studying and culturing microorganisms. And we're going to start with microscopy. This is not a physics class, so you really don't have to know a lot of let's call it, effectively physics stuff, okay? Such as reflection and refraction and things like that. What I want you to understand in terms of microscopy, light microscopy as a method, is that, first of all, we do use refraction and reflection of light. We use a system of mirrors and lens. And using the system of lens, we can enlarge the image that we're looking at so we can see something that can't be observed with the naked eye. The microscopes that you're going to work with, not today, but next week, actually, are light microscopes. It does not reflect their weight. It reflects the fact that they use light to visualize the specimen. They are compound, which means they have many lenses. They're binocular. They have two eyepieces. And importantly, they are parfocal. The term parfocal refers to the fact that when you change objective lenses, let's say you go from 4x lens to 10x lens, the image remains in the relative focus. Three main characteristics of any microscopic observation are magnification, resolution, and contrast. So let's define those terms. I will ask you the definitions, the determinations of those three terms. So first of all, magnification is the ability of a microscope to make things larger. When you work with a microscope, you can calculate the magnification that you are working with by multiplying the magnification of eyepieces by the magnification of objective lenses. So for example, eyepieces on the microscopes that are in the lab are 10x. If you combine that eyepiece with the objective that has a magnification of 40x, the actual magnification that you impose on the image are 400x. Clear? Excellent. Resolution isn't the same as magnification. It's ability to see fine details. I bet most of you watched TV shows like NCIS or, CSI, stuff like that. And you may have seen circumstances when there's a camera shot of a license plate that looks awfully blurry, and then they enlarge it and enlarge it and enlarge it, and out of a sudden, this image becomes super sharp. It's not going to work this way. Just not. Resolution depends on the wavelength of light. It depends on many other things. But for our intents and purposes, we want to focus on the wavelength or the frequency. So I don't want to go into the physics of how the wavelength determines resolution, but let's put it this way. Visible light has a limit of the size at about 0.3 micrometers. Don't memorize this number. I will never ask you this number. What I can ask you is what limits the resolution? And the answer is the wavelength or frequency, which are basically the same. Now, what about contrast? So contrast is not a tricky thing, but I have a question I want to ask you. Which molecule, which chemical compound is the most abundant chemical compound in any cell? I'm going to give you a hint. It's a very simple compound. Any ideas? It represents about 60% of any cell.
Oxygen?
Nope. Nope. Compound, not element, huh? Who said water? There you go. It's water. Okay? Cells are mostly water. When you look at cells under the microscope, and I'm speaking about bacterial cells, you have to immerse them in water. So in some way, you look at the sacks, bags, call it whatever you want, containers of water, mostly, in the water. It's not going to provide you much of a difference in terms of the contrast, in terms of the density between the cells and the background. So contrast is the difference in the light absorption between the cells of your specimen and the background, which is usually water. We're going to discuss how you can increase that parameter, that contrast. Okay? First microscope was developed by Anthony van Leeuwenhoek. I cannot help myself but to brag that I had one of the most nerdiest privileges. I actually visited a tomb of the guy. It was like really cool for me. I know it's weird, but I saw the tomb of Anthony van Leeuwenhoek. Okay? So his first microscope was fairly simple. You had a magnifying glass, and the specimen was placed over here. What do you think he looked at first? Like he was basically a guy who made glasses. Microscopes was his side gig. What did he look at first? What do you think? No. What I'm saying is, what was the sample? Like he developed a microscope. What was the first sample that he looked at? Nope. What? Bacteria. Okay. Well, he didn't know about bacteria. What did you say? Huh? Food. No, not food. No. Say again? No, he didn't know about cells. Come on, you are a male. You want to look at something. Hold your horses, not semen. Thesis. The best answer, yeah. He actually observed Giardia lamblia in his feces. He was infected with Giardia. The best answer I got, though, was penis. My only response was it that small that he needed a microscope to look at it. Anyway, later his microscope was improved. So effectively this is the first compound microscope that was developed by Robert Hooke, British scientist. He also was the first to actually observe cells. He didn't know that it was important, just looked at the cork and thought, oh, okay, cool. So these are the parts of a microscope, eyepiece, the part that you look into, objective lenses with a revolving nose piece that, you can change objectives, stage where you put your slide on, focus knobs, coarse and fine. We're going to talk about the function in the lab. It's just, you adjust focus. We talked about stage. Stage knobs to move the stage around. Now, few things that may cause some kind of, trouble understanding. Rheostat. It's a little dial that changes the intensity of light. Illuminator is a fancy word for light bulb. Condenser is special lens that focuses light on the specimen, so it does not spread out like this. And finally, diaphragm is the device that, don't ask me the physics. I have no idea how it works. But diaphragm additionally allows you to regulate the amount of light that reaches the specimen, but it also changes the resolution. Surprisingly, less light means better resolution. Any questions so far? So I think I mentioned it in last week that if you don't see something in the notes and in the study guide, it's not going to be in the exam, and I'm going to hold true to my promise. So these are 7 types of light microscopy that you must know. And I'm going to give you a brief outline of what they are, and I'm going to give you an idea what type of question you can expect in the exam about those. So the type of question you can expect is effectively matching the term for that type of microscopy with its description of features. It can be when you use bright field microscopy, you are, and you pick one of the answers. Does that make sense? Or you can have a question something like, which of the following statements about confocal microscopy is correct? Okay? So let's give simplified definitions. Bright field. The cells are dark, stained, colored, whatever, and the background is not. Okay? Does that make sense? So the background is bright, and the cells are observed at the bright background. Dark field. The opposite. Okay? So cells are highlighted. Cells are illuminated through certain optical trickery, while the background remains dark. This type of microscopy, for example, is used for microorganisms like Borrelia burgdorferi that you can see on this image. Borrelia burgdorferi is one of the spirochetes. They're super thin, and it's really hard to see them using bright field microscopy. So brightly lit cells against the dark background. Phase contrast. When you add more optical trickery, diffraction, interference, you can observe living cells in their natural habitat with a high contrast and high resolution images. It's important you can take those pictures on the live specimen without staining, without treating cells with any kind of chemical. DIC, or differential interference contrast microscopy, is basically phase contrast that is taken up a notch. So if you look at this image, you can see that cells, you kind of can appreciate a 3D shape of the cells. Do you see that? So high contrast, three-dimensional appearance within the live, unstained specimen. So we'll see why I focus so much on live and unstained. So you effectively can mix bacteria with water, subject them to DIC microscopy, and observe individual cells with their 3D shapes. Okay? Now, fluorescent microscopy. Some cells, I'm going to step back for a second. You familiar with black light? That's it. You use UV light to make things fluoresce. Never bring a black light into a hotel room, by the way, human sperm does fluoresce. It's going to be everywhere. Anyway, some cells fluoresce naturally. For others, you can have special dyes. Okay? So this is Pseudomonas putida, a microorganism that you can stain with a specific fluorescent dye, and it will, well, fluoresce when you expose them to black light. The advantage of this method, there are reagents, let's call them this way, that will bind or stain only a certain type of cells. Do you follow my train of thought? So for instance, you can have a stain that will only bind to bacteria called mycoplasma. So when you look at your sample and something fluoresces there, you know it's mycoplasma, not anything else. Clear? So it can be made selective. Confocal microscopy. To be honest, this is one of the worst confocal microscopy images that I've seen in my life, if not the worst. If it is used properly, confocal microscopy is quite awesome. So here's what it does. You use a microscope, confocal microscope, to take multiple, so I'm going to tell what a Z-plane is. Okay. I think I lost the charge. Give me a sec. I'm going to try to charge it. In the meantime, I'm going to use my mouse, and I'm going to try to charge my pen in the meantime. Okay, so we can highlight using the mouse. So multiple Z-planes, right? What does that mean? You focus on different parts of a specimen and take an image. You go deeper, you go shallower. Does that make sense? You effectively are doing a tomography. Do you follow my train of thoughts? So you take slices of your image. And then special software combines those slices to produce a three-dimensional image of your specimen. Does it make sense? It is effectively tomography. So multiple shots in multiple different focal planes to produce a three-dimensional image. And finally, to photon microscopy. You can presume, and you will be quite accurate, that when we go from bright field all the way up to photon, if you think about the price of equipment, it increases kind of exponentially. So I worked with practically all of them. If you want to do fluorescent microscopy, the microscope and all the bells and whistles running about 10 to 15 grand. Confocal, when I was working, it was 50 to 60, now it's probably more. 2 photon, the setup that we had in the department was $250,000. Okay? It's awesome because you can put not just a specimen, you can put a live specimen like a mouse muscle, part of a mouse, under the microscope. And within that muscle, you can distinguish different cells, and you can use a variety of fluorescent labels to color them differently. So you can color, let's say you want to look at the response of the mouse immune system to the bacterial infection. You can stain macrophages, green, and neutrophils red, and T cells blue, and you can see them all together, real time, moving around, dealing with that bacterial infection, actually making videos. This is a pretty good stuff, okay? Now, I want to highlight that all of this is the type of microscopy that uses light. And as I mentioned before, light microscopy has that resolution restriction, remember? Because the wavelength is limited. The limit of light microscopy is 0.3 micrometers. I said it before, don't memorize the numbers. It's not a number I want you to know, just there's a limit. So what if you want to look at something that is much smaller, i.e. viruses? You can use, instead of light, you can use a beam of electrons. When you think about light, actually I have a question for you. It is a legitimate question. When you think about light, do you think about light being a bunch of particles or light being a wave? Wave? Say again? Yes. 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 The de Broglie paradox. Yes. Light is a wave and a bunch of particles. Okay? So we're not going to get into quantum physics of that. But I want you to acknowledge that light is being a bunch of particles, and those particles, they reflect, they refract, they fly around, right? So same can happen to electrons. Electrons can be reflected, can be refracted, okay? So when you target an electron beam on your specimen, you can effectively produce two types of images. One of them is the transmission electron microscopy right here. In this case, the beam of electrons goes through a very, very thin slice of your specimen. Am I making sense? What you're seeing here is one of the most famous images in virology. It was taken by Fred Murphy, I think Fred, his name. It's a first image of Ebola virus, okay? And I call it a Mickey Mouse virus. But the idea is you can see that there's a very dense layer on the outside, you know, this dark line, and lighter content within the virus. But it only gives you the information what's in there. Do you follow? Scanning electron microscopy, on the other hand, allows you to identify the shape of a cell or a virus, whatever you're talking about. Am I making sense? Now, if you look at these Campylobacter cells here, you can see the shape. You can see that they kind of rods that a little bit wavy, you know, there's some flagella present. Does that make sense? You have no idea what's inside. Okay? Does that make sense? Yeah. So it's like, you know, you can look at the stick of salami and acknowledge how long and how thick it is, but you don't know what's inside. If you look, that's scanning electron microscopy. If you look at the slice of salami, you know what's inside. You don't know how long the stick is. Does that make sense, the difference? So I will ask you to match the description of one of these overall 9 methods, 7 for light and 2 for electron, and the name. Okay? Also, keep in mind sort of applicability. Remember how we were talking about the fact that if you want to observe viruses, you're probably going to need electron microscopy. So if I ask you, generally speaking, if you want to look at the shape, outer shape of the virus, think, okay, virus, that's electron. Outer shape, scanning electron. Making sense? Okay, let's move on. So one of the problems that for us remains somewhat unresolved. Let's see if is going to work, and I don't know what's going on here. No, but I'm probably going to return this pen eventually. It's kind of a, right, we'll figure it out later. Never mind. So, I'm going to use the mouse for now. When you prepare your specimen, and we're going to work with viral specimens for the most part, you usually do it through the wet mount. Okay? So, you take your bacteria, mix it with water. That's wet mount. Good? Well, I would argue that it's not good. Because in that wet mount, cells are going to move around, and if you try to stain them, you're going to wash them away. So you have to do several things. You have to immobilize cells, and you have to make them immovable. You have to make them sticky and ideally dead for the purpose of staining. This can be achieved by the fixation. Fixation is the process that kills bacterial cells and also makes them stick to the surface of the microscope slide. Okay? You can do it using chemicals, which is very inconvenient, or you can use heat. On this illustration, you can see, heat-mediated fixation. So you effectively make a wet mount, put the slide with the wet mount on the hot plate and wait for it to dry. When it dries out, cells die and become fried to the surface of the slide. We're good so far? So now they are dead and demobilized. Now you can stain them. The main goal of staining is to increase the contrast. Cell used to be colorless, and suddenly it's, purple. Okay? So stains can be classified using two approaches. First approach is simple versus differential. Simple stain is the one in which all cells, no matter what they are, appear the same. So for instance, if you make the slide which contains a bunch of different cells, fix it and then cover it with crystal violet, all cells will appear violet. Clear? Differential stains, on the other hand, allow you to distinguish different bacterial cells based on their structural differences. For example, cells that do have capsule versus cells that do not. Cells that do have flagellum versus cells that do not, and so on and so forth. Okay? Another classification that we can make for stains is the positive or negative. Let me change it to the highlighter. So what are positive stains? When we talk about positive stains, it means that the stain actually binds to the cells and stains the cells. When we're talking about the negative stain, it means that the stain remains in the background. Cells themselves aren't stained. So we need to figure out why that happens. Before we proceed, I have a relatively simple question for you. The charges of opposite signs, positive and negative charges, how do they behave, how do they interact with each other? See, attract, right? Opposite charges attract. And charges of the same sign repel each other. Good? So, if you go back to our conversations about the structure of bacterial membrane, you'll notice that it is composed of phospholipids. Phospholipids have a negatively charged head. We're on the same page, folks? All good? So, overall, the surface of bacterial cells, it carries a negative charge. If the chromophore compound that you use to stain, if it is charged positively, it will be attracted to the negatively charged surface of bacterial cells. Reasonable? So, positive stains have positively charged chromophores that bind to the negatively charged surface of bacteria. Negative stains, on the other hand, have negatively charged chromophores. They are being repelled from the negative surface, negatively charged surface of bacterial cells. They remain in the background, and you can see stained background, but the cell remains unstained. Does that make sense? Okay. So now, what are main differential stains? You must know. First is the Gram stain, which allows you to distinguish between gram-positive and gram-negative cells. I know that you may probably have no idea what it is. Give me 3 minutes, we'll get back to this, okay? Acid-phos stain, same story. We're going to get to acid-phos stain a little later. I want to highlight these three. Endospore stain first. Last week we had a conversation about different structures of bacterial cells. Do you remember that? The endospore, the structure that protects genetic information. You can stain for it. It's an important tool because the number of species that can produce endospore is quite limited. So, first of all, no pictures on the exam. Don't worry about that. Here you can see in this image on the right, endospores are green and the vegetative cells are red. Okay? Then we have flagellar stains. They stain well, flagella. The mechanism is super interesting. I love it, the mechanism. What the stain does, flagella are very thin. They cannot be seen with a light microscope. What the stain does, it makes flagella literally thicker. The staining molecules just bind to the flagellum. Okay, making it thicker, making it visible. All good? And finally, the capsule stain, the white halo that you can see around each cell on that image are capsules. So the good news, I will never ask you questions about capsule, flagellar, or endospore stain, because the answer will be in the question. I can't ask you, what is the fun? What do you use endospore stain for? Duh. Right? Now, let's get back to these guys. Gram stain and acid-phos. So remember our conversation about the structure of bacterial cell wall? Yes? There are gram-positive cell walls and gram-negative cell walls. So microbiology has it kind of backwards. Now, listen, it's my little pet peeve. Hear me out. Fundamentally, most bacteria can be divided into gram-positive and gram-negative. Remember gram-positive having a membrane and a thick cell wall, and gram-negatives having a membrane, thin cell wall, and outer membrane. The reason why one is called gram-positive and another one is called gram-negative is because in 19th century, the guy named Hans Christian Gram developed a staining technique. And the staining technique revealed that there are two very distinct and very large groups of bacteria, gram-positive and gram-negative. The main difference between them is the structure of the cell envelope. Okay, does that make sense? Why do you think this distinction, gram-positive versus gram-negative, is important? In other words, I'm a physician. I treat a person with a bacterial infection. I want to know whether the bacterium that affects that patient is gram-positive or gram-negative. Why do I want to know that? You don't have to raise your hand, just yell.
Because gram-positive, the cell wall is thicker, so it's harder to treat than gram-negative. And then certain antibiotics, they treat different
There you go. Yes.
bacteria.
Different antibiotic choice. Does that make sense? I'm going to give you an example from maybe your own experience. Anyone had, well, everybody had, sore throat, strep throat. Do you remember what you got? Lucky you. Anyone remembers what you guys got prescribed for strep throat? Okay. Amoxicillin, thank you very much, or penicillin. Amoxicillin and to a great extent penicillin can only kill gram-positive microbes. Strep is gram-positive. Now, next question, please don't answer. I mean, you probably have an answer in your head. I don't care about your answer. Urinary tract infections. If you were unlucky to have one, the antibiotic that you get prescribed when you're diagnosed with UTI is usually third-generation cephalosporin. There's a reason to it. Do you want your patient with UTI to wait until you identify what they have?
No.
Probably not. The 70% of all urinary tract infections are caused by gram-negative E. coli. Second in line, gram-positive Staph aureus. Okay? Does that make sense? You want to cover all bases here. Third-generation cephalosporin is a broad-spectrum antibiotic that works equally well against E. coli and Staph aureus. Does that make sense? If it doesn't work and your lab goes back and people, those folks know what they have on their hands, they may adjust antibiotic, making different choice. But this is why it's important to know whether the infection that you, as a medical provider, infection that you're dealing with is gram-positive or gram-negative. Question? No? Okay. So that's why it's important. What about acid-fast staining? So acid-fast staining is used for mycobacteria. We talked about them as well. They have that waxy layer that renders gram stain useless. Okay? So you would use acid-fast stain for mostly Mycobacterium tuberculosis, maybe Mycobacterium avium, some other species. But the point is, you reserve this stain for bacteria that carry mycolic acid. Sounds good so far? Everybody? Right? Let's move on. So, gram stain. You're going to do it in a couple of weeks. I want to talk about how it works. Let me make another futile attempt on. I am returning this. I don't like this. So, what's going on here? When you do gram stain, you have, let's say you have a mixed sample, right? Gram-positive, gram-negative, whatever, right? And you cover them with crystal violet, which stains all the cells, gram-positive, gram-negative. It stains them purple. All good? All right. Then you have step 2, iodine. So crystal violet is called a primary stain. You need to be able to match the term and the purpose. Then you treat your cells with iodine. The term for iodine is mordant. So what it does is making crystal violet sticking to the cell wall even better. Which cells have a thicker cell wall? Gram-positive or gram-negative? So overall, that crystal violet dye sticks to gram-positive cells better. Right? And then you apply alcohol, which serves as decolorizer. So decolorizer can remove crystal violet from the cell wall. But crystal violet is stuck to gram-positive cell wall and not so much to gram-negative. Am I making sense? So if you decolorize properly, you're going to remove crystal violet from your gram-negatives, but not from gram-positives. Are we good? Right? So now your gram-positives are still purple, it's all good. Gram-negatives, bless you, gram-negatives are colorless though. You need to color them by using a counter stain known as safranin. Safranin stain cells pink. Your gram-positives are purple, so the addition of the pink stain doesn't change their appearance. They're too dark for that. But your gram-negatives that used to be colorless are now stained pink. Does that make sense? This is effectively what you're going to do in 2 weeks from now. Doing a gram stain, I'm going to make you mix gram-positive and gram-negative microorganisms, and you must, identify them separately. You must see both of them on your slide. Okay? We're going to do gram stain, we're going to do acid fast stain, and we're going to do endospore stain in a lab. So since we mentioned endospores, I can't help myself but talk about one of my favorite bacteria, which are bacilli. You may remember from the last week's conversation that the term bacillus describes a microorganism which cells are shaped as rods, right? So that term was used as a genus name for bacilli. I'm going to highlight some either clinically or socially important species. Scientifically important, most important is Bacillus subtilis. Among all gram-positives, it is the most studied gram-positive microorganism. It's not an easy question, but maybe you can guess. What is the most studied gram-negative microorganism? You 100% know the name. If we say bacteria, what is the first name that comes to mind?
E. coli.
There you go. The most studied gram-negative bacteria, E. coli. Okay. Interesting bacillus called Bacillus thuringiensis is used as a source of an insecticide. It's called BT. What's actually cool about this, it is certified organic. We can talk about my opinion about organic foods some other time. Just, it's not positive, but whatever. What's cool about BT is that it affects only insects. It does not have any effect on humans. So in the places like Florida, especially the southern tip of the flaccid penis of America, they use BT as an insecticide to control the population of mosquitoes. Okay? Now, these guys are not pathogenic for humans. These two are. First is Bacillus cereus. Bacillus cereus per se itself is not a pathogen, but it produces a toxin. So how do people get sick from it? Remember, bacilli, they form endospores. Okay? So it turns out rice contains a whole bunch of endospores of Bacillus cereus. It's an environmental microorganism. Okay? Now, if you are a restaurant owner and you cook rice, you're not going to cook an individual batch for every client that orders it, right? You're going to have some kind of a big pot, rice cooker, whatever, that maintains rice at the warm temperature. Warm temperature, humidity, carbohydrates from rice. It's a perfect environment for endospore to germinate. And so it does, and produces vegetative cells of Bacillus cereus, which grow, produce toxin, and then die because it's a bit too hot. But toxin remains in rice. And then people consume that rice and get food poisoning. It's not horrible, it's like 2-3 days. Like you can tough it out. But it's been shown that food poisoning incidents associated with the rice served like this, like I described, are heavily associated with the toxin from Bacillus cereus. Okay. Now, Bacillus anthracis is pretty cool. This is the closest we got to developing a biological weapon. Biological weapons make absolutely no sense. Good old nukes work way better than that, more reliable, more controlled. Okay. So, Bacillus anthracis is the infection that affects, for the most part, cattle. Well, not only cattle, let's put it this way. Hoofed animals, ruminants. So cattle, it's pretty common among reindeer. When these animals get infected, humans can acquire the disease by the physical contact with animals. So you see the scars, images 2 and 3, right? So those lesions are cutaneous anthrax. It looks horrible, it is super treatable, and it heals. Okay? Much worse situation is when somebody eats an animal that is sick. Okay? This is enteric. It can be very fatal. Okay? But the worst one is pneumonic anthrax, which is the result of the inhalation of the spores. It's the most severe, super lethal. Most of you were not conscious at the time, but in 2001, after 9/11, there was a series of anthrax terrorist attacks where spores were sent to different locations in the United States. Turns out it was domestic terrorism. There was a guy from CDC who was like just disgruntled. Okay? But several people died because it was inhalation. Make sense? So anthrax is an example of zoonotic disease that comes from animals. Doesn't really cause, there's no transmission from person to person, realistically speaking, it's animals to people. And one of the biggest problems in controlling such a disease, so imagine, I'm going to ask you some questions. Just pretend that you are solving the problem, all right? So you have 100 cows that are infected with anthrax. Remember, you cannot eat them. It really can kill people. So what are you supposed to do with them? Say again? Butcher, yeah, well, it's called calling, but yeah, kill them. What are you supposed to do with corpses?
Bury.
Okay, ideally, you will bury them, we'll get there, but ideally, what are you supposed to do with that? Burn them. We burn humans, come on. Like, we burn humans all the time. The only difference is that you need a bigger stove, oven, right? And last time I heard, first, the two problems. Cows are big. And problem 2, they're usually not hanging around the crematorium. So I don't know if there's a business I would do a startup like mobile crematoriums, just drive to your house and just burn there, you know. But there are no mobile crematoriums for cows. And burning them on a pile of wood isn't going to do the job. So yes, people have to bury them. So they dig a hole in the ground, put them in, cover them with some chemicals, put the dirt on top of it, and mark the territory that it's not safe to use. Eventually, though, usually signs go away, and these bacilli form endospores, right, folks? And endospores are really resilient. And they will withstand all those chemicals and shit. And eventually, capillary forces, groundwater, will get the endospores on the ground, and there will be another herd of cattle eating, getting infected. This is inevitable. That's going to happen. If you look at the map on the bottom, the yellow color shows you sporadic outbreaks, so from time to time. We do have anthrax in the United States. There are countries where it's pretty common, but we do have anthrax. I really wonder why Egypt doesn't have anthrax. Like seriously, I don't know. I understand why northern countries don't have anthrax. They don't have animals, pretty much. It's so fucking cold. But it's, you know, it's kind of everywhere. And it's not vaccine preventable, by the way. All right, now we're going to jump into the process of culturing microbes. I'm going to give another try, desperate try, to be honest, to this. No, it's so sad. I'm going to write a very angry email. Whatever. So, culturing microbes implies 5 steps. You don't have to write them down because we're going to go through each step. Inoculation, incubation, isolation, inspection, and identification. So we're going to start with inoculation. What does the term inoculation mean? It means deliberate placement of your bacterial sample on the agar, in the medium. Okay? I highlight the word deliberate. Remember last week you were sampling the environment? You had an intent. You collected a sample and you put that sample on the agar plate. Right? You didn't just sneezed into a agar plate accidentally. Like that sneeze is not inoculation, that's contamination. Inoculation is deliberate. Second thing, medium. What is medium? Medium is anything that can support nutritionally the growth of microorganisms. So the agar that you used last week was obviously a medium. But let me ask you a few questions. Milk, can it be a medium for bacterial growth? Yeah. New England clam chowder. Sure, why not? If nutrients are suitable, sure. What about me? Can I be medium? Of course, all the time. Bacteria grow on me, in me, so yes, I'm a medium. Whatever it is, what about that? Unless there's something that can legitimately break down plastic for nutrients, no. This cannot support the growth. Does that make sense? And I want you, like, my goal, one of my overarching goals when I teach micro, is first of all, gross you out. Second, ruin some foods. Third, appreciate the importance of microbes. And fourth, understand that some surfaces, they don't really have microbes. So let me ask you this. Do you think this wall right here has any microbes?
Yeah.
What would they eat? So again? What would they eat?
Stuff in their ear.
Come on. Like, it's not magic. They can't eat stuff, and there's not a lot of stuff in there. There's nothing to eat. They probably all die over there. And people don't touch it over and over again. Does that make sense? So, like, if you're in the hospital and you look at the doorknob or faucet or things that people touch, sure, there's going to be plenty of bugs. We had students sampling these grills on the vents. They couldn't isolate anything from them. There's, they dry, there's no water, there's no food. Bacteria cannot survive. So when you think, oh, everything is contaminated to an extent. Okay? So medium is to be able to support the growth. Inoculation is a deliberate process, and it goes without saying, you have to use sterile instrument to inoculate. You don't want any contamination. So medium can be classified using several approaches. First classification is based on its physical state. So liquid, semi-solid, and solid. So you already used solid medium, agar. It's solid at room temperature, and when you heat it up, it liquefies, it melts. When you cool it down, it solidifies again. It's very convenient because you can mix the agar with whatever chemicals you want. You can change the composition. You can boil it, sterilize it, cool it down. You have your tailored medium. That makes sense to you? What would you use agar for? The solid medium. First, colony isolation. We're going to see some bacterial colonies today, but you probably saw them before in some other bio courses. Okay. So it's useful to distinguish between different bacteria. You can't really grow a huge amount of bacteria on agar. You can also use agar to characterize bacteria in a way like, do they ferment glucose? Do they not ferment glucose? Do they break down starch? Do they not? And so on and so forth. Okay? So far, so good? The opposite to this is liquid medium, which is, well, liquid. Liquid media have two main purposes. Purpose number one is characterization, tests. If you look at these guys here, you see the red, super red on the left, and well, I shouldn't have used that color of the highlighter, but you get the gist. Orange on the right, right? Orange color represents low pH, which means there's fermentation. That's your indicator medium. You can also use liquid medium to accumulate insane amount of bacteria if you want to. So like you just need a bunch of bacteria, right? So you inoculate like 2 liters of medium, and then you have 2 liters of bacteria. There you go. Okay? In between those two stands semi-solid media. It's a clot. Doesn't have a solid surface. And I compare it to this. If you will take a tube with semi-solid medium and flip it upside down, eventually, maybe in a couple of weeks, it's going to crawl out and fall out. Because it's not really super solid. Does that make sense? What do we use semi-solid media? Also known as slants. See, slanted shape. Two main applications. It can be for tests. Like this is the what's called triple sugar iron agar. Okay. I'm not going to go into the details, but you clearly see that 4 tubes here have different appearance, right? It's because different bacteria, well, the rightmost tube is not inoculated, but 3 different bacteria in the first 3 tubes have different properties. Okay? Like fermentation is in first tube and H2S production in the third. So you should be able to match the description of the medium and the purpose and the term in terms of the physical state. So like we use it to separate colonies, that's solid. We use it to grow a shitload of bacteria, that's liquid. Make sense so far? Medium can be classified based on its composition, synthetic versus complex. So how do we make a synthetic medium? We take water, fixed amount, hear me out, fixed amount of water, and we add fixed amount of every amino acid, fixed amount of glucose, fixed amount of salts, right? So we know the exact chemical composition. We define the chemical composition. Do you follow my train of thought? Like if you ask me what's the concentration of amino acid aspartate in the medium, I can tell you right away. Do you see what I'm trying to say? Now, defined medium is convenient because we can control for components. Like I want to say, I wonder if my bacteria can grow without aspartate. I can take all aspartate out and see if it happens. Clear? Complex medium does not let you do such a thing, but its chemical composition is unknown. By unknown, here's what I mean. Remember we were talking about milk being a medium. Okay? If you buy milk, grocery store, do you know the exact chemical composition of milk? Like all the grams and micrograms of every chemical that is there? You don't. Or even better example, you cook soup at home. Just make some, New England clam chowder. Do you know the exact chemical composition? You don't. However, you know that it has plenty of nutrients. It can support bacterial growth of very different bacteria. This is why complex media, I usually compare them to a buffet. In terms of restaurant, like you go into a buffet that you don't know, you don't know which exact dishes are going to be there. But you know that you're going to find something that you will eat, right? Defined media are like a la carte. You look at the menu, like, I'm not going there. Okay, does that make sense? Nothing for me here. Okay. Questions on the chemical composition. I'll give you a summary of a question at the end of this conversation. General purpose. So general purpose enriched selective differential. So what does that mean? General purpose. I'm kind of stumped. It's self-explanatory, isn't it? The agar that you used last week, trypticase soy agar, that's a classic general purpose. It can grow virtually anything. Now, I use the word virtually. There are some bacteria that are picky. We call such bacteria fastidious. They need something special to grow. For example, members of the genus Neisseria cannot grow without a significant presence of iron in the medium. So if you want to grow Neisseria, you will use blood or chocolate agar. Both types contain blood. In one case it's fresh, in another case it's cooked. Okay? But overall, this medium is enriched by iron. Do you follow? Allowing fastidious bacteria to grow. Common confusion that students have, they confuse the terms enriched and fastidious. Those are not the same. Fastidious means picky, and enriched means, well, enriched. Sounds good? Okay. Selective. Inhibit some bacteria, allow other bacteria to grow. Pretty straightforward. Differential. Microorganisms that have different properties appear differently on such medium. So far, what I said, does it make sense in terms of definition and explanation?
So for the purpose, is that for the complex?
All of them. So, thank you for this question. I want you to understand that the classification based on the physical state, on the composition, and on the purpose, they can overlap. They are independent. And I was actually about to give you an example of a question. So here's my question. And listen, an EMB agar, you don't need to know what EMB is. EMB agar, contains chemicals that selectively inhibit the growth of gram-positive bacteria. Okay? It also contains lactose. And bacteria that can ferment lactose appear differently from bacteria that cannot. What is the proper description of that medium? And you start thinking, okay, here, well, it's not a write-in. Remember, you will have to choose out of multiple choice, but agar, solid, right? Inhibits growth of gram-positive but not gram-negative. Selective. And allows you to distinguish lactose fermenters from non-lactose fermenters. Differential. Does that make sense? So you look at the list of answers and you're like, solid, yes, checks out. Selective, checks out. Differential, checks out. Boom, that's your answer. Does that make sense? Obviously, I can, okay, this medium contains, I don't know, brain-heart infusion, agar. Brain-heart, you don't really the chemical composition, so it will add to this that the medium is complex. Okay? Now, I strongly encourage everybody, when you're going to have an exam next week, and you read the questions, and you have the questions about the questions, you come to me. I don't want to hear from you, oh, I didn't know what you meant by this. My goal is because yeah, I may phrase it weirdly, you know, but please do not hesitate to come and ask. Okay? All good? Moving on. Any questions? Cultures, 3 types: pure, xenic, mixed, and contaminated. Pure or xenic cultures mean that they consist of one species. Okay, it's pure. Mixed, you know that there's going to be 2 or more species. You collected samples from the environment, you probably can expect more than 1 species. Contaminated, you wanted 1 species, but you got more than that because you did not do a good job with inoculation. Does that make sense? Mixed and contaminated are not the same. Mixed are expected, contaminated are not. Cultivation of viruses. We briefly mentioned that viruses can only reproduce inside of the cells, so you have to grow viruses in the cells. In these flasks that you see here, these flasks have cells on the bottom, like that surface, okay? That makes sense? Cells grow there in their own special medium, and viruses infect cells, so cells effectively are media. You can use live organisms as media. So this lady inoculates eggs to produce flu vaccine. Still makes some flu vaccines from using eggs. There are some organisms actually that use animals, okay? You obviously can grow viruses, but you can also grow viruses in cells. You don't need animals for that. But there are some elements. For example, Trichinella spiralis. Any hunters here? Ever heard about Trichinella? Yeah? Which wild game you're not supposed to eat so you can avoid Trichinella? Say again? Bears, and change 1 letter in the word bear. Boar. Bear and boar. Unless you cook them really, really well, to the extent that they become like rubber, you should not eat either. Okay, boar is a little better, like it's pretty much pork. Okay, but bear is dog. So trichinella is a helminth, it's a worm, that you can only grow in mice in the lab. So you effectively have to infect mice and then use them so they accumulate a lot of worms, and then you sacrifice mice and basically like do a mouse smoothie and treat it with some acid, and you isolate worms. Okay, you can use plants for plant viruses, especially. It's really hard to make separate cells, plant cells, to grow, so it's just easier to infect actual plants. Okay, so hear me out. Now we inoculated our medium. Next thing that we need to do is to incubate. You already, quote unquote, met the incubator last week, 2 of them actually. One maintains 25 degrees Celsius, and now there's 37. Well, there is a temptation to say that we need incubation because it's warm, but no. We can incubate cells at any temperature we want, pretty much. The idea behind incubation is that it makes your results reproducible. Okay? If you incubate your cells at room temperature, and I do the same, we may have different room temperatures. But if you incubate at 37 and I incubate at 37, that's the same 37. We can compare results and we can reproduce this result. I'm not arguing that incubator allows you to form ideal environment, right? Like optimal temperature, optimal gas concentration. Some bacteria require a certain amount of oxygen or carbon dioxide. Okay, all good? Now you completed inoculation, you completed incubation, you get your results. But I have a question. If you isolate, if you take a sample from the environment, right, and you look at your plate and you see colony, it looks pink. The colony color is pink, okay? You have to ask yourself, is it really pink? Or I have some bacteria that appear white, some bacteria that appear red, but they grow next to each other. So altogether, it looks pink. So what you got to do to make sure that the species that you're working with is actually single species, not a mixture. For this, we use isolation. Now, if you look at this, this is what's called a streak plate. So this bottom one is the first streak, okay? So think of it this way. You have a big slice of bread, and you put some butter on one end of that slice, and the layer is pretty thick, okay? Then you wash the knife, and you spread small amount of that first chunk of butter over the next, I don't know, couple of inches of your big-ass slice of bread. Clear? And you keep going, you're effectively diluting each streak further and further, so eventually you start seeing individual colonies. Does that make sense to you all? Okay? This is why we call it an isolation. We are actually going to practice the streak plate. And you will use mixed culture, and your goal will be to isolate 2 different species that look very much different, on your plates. Okay? Now, I love streak plates. They're so intuitive, they're so telling, but it's not the only way to isolate. You can also do pour or spread plate. So imagine you have liquid culture of bacteria, which is mixed. Hear me out, mixed. You know it is. So what you do, you make serial dilutions of that original culture. Do follow. The more you dilute, the less bacterial cells you have in your dilution, the lower the concentration. Agreed? So when you start plating these dilutions, you get, okay, I'm sorry, I don't know, don't ask, don't tell. So when you start, no, it does not respond fine. You start seeing less and less colonies. And eventually you separate them enough to be able to identify whether you have one species, or two species, or three or four, whatever. Am I making sense? Now what's the difference between the pour plate and the spread plate? There are two approaches. One is to make serial dilutions of bacteria in the agar. Remember, if you heat up the agar, it's going to melt. It melts at a relatively moderate temperatures, between 48 and 50 degrees Celsius. And if you make dilutions quick and pour them, this is why it is a pour plate method. If you pour them, they cool down fast enough so bacteria don't really die. Okay? Another approach is to make the dilutions not in the agar, but in the liquid medium. Then you apply some of that dilution on the plate, and using that little device that you can see there called a hockey stick. This is what it's called in every lab. It's a hockey stick. You spread your sample on the surface of the agar. Good? And then same effect. So we've covered 3 I's. We've covered inoculation, incubation, and isolation. Now we are to the inspection. Inspection is really simple. Inspection includes the analysis of your colonies. How they look? They're big, are they small? They're large. Are they round? Are they irregularly shaped? Their margin, is it entire? Is it wavy or undulated? Is it rhizoid? Okay, so you basically provide a set of visible characteristics. Inspection also includes microscopic evaluation of your microbes. So, hear me out. Let's say I have my culture, and let's say I culture my throat. Let's say I have a sore throat. I don't, but let's say. And I collect the culture and I put it on an agar, and what I see is a bunch of relatively small, round, white colonies with the entire margin. There's a bunch of bacteria that will produce such colonies. Doesn't tell me much. So what I do next, I take some of those bacteria from the colony and I put them on the slide and I fix them and I do gram staining on them. What I see now, cells are round-shaped, cocci. And they are arranged in chains. The chain of cocci is streptococcus. So it is a strep. Which strep? I don't know. Can be Streptococcus pyogenes. Can be Streptococcus pneumonia. I don't know. But at least I narrowed down to Streptococcus. Does that make sense? So inspection can guide you towards a certain conclusion, but will not provide the exact answer for the most part. Am I making sense? Which brings us to the identification of microorganisms, and we're going to discuss 3 main categories of methods. Immunological, biochemical, and genetic. Well, for the love of God, maybe it will help, but it will not. It's annoying. Okay, fine. So we're going to start with immunological methods. Since the product of Chinese industry failed me, I will have to talk. Okay? So immunological methods are based on the interactions between an antigen and an antibody. Who did not take A and P2 yet? Raise your hands. It's not, I'm not, okay.
I'm in it right now.
Okay, right now, yeah, okay. So basically, when you have some kind of an antigen, like a protein, and you stick it in a foreign species, right? You follow? The foreign species will produce antibodies. Antibodies are very, very specific to that antigen. So we can raise antibodies to a variety of antigens, and they use those antibodies to discover the antigen. I want to highlight three methods. First is, and I absolutely love this method, is a Western blot. So in Western blot, or the proper name of it is immunoblot. Let's say you have a sample of patient's blood and you suspect that there is an antigen in the blood. Antigen that belongs to some virus. Are you with me? Blood contains a lot of proteins. So you can't really parse out the signal from the whole blood sample. You need to separate the proteins. So you load the sample onto a gel, which effectively a lattice made out of the polymer called acrylamide. So proteins, and then you apply electromagnetic field, and proteins start running through the gel. Are you with me? Which proteins move faster, small or large? Small, as my professor of protein chemistry told us. Scrawny kids run between the trees and the fat kids get stuck between the trees. Okay? So large proteins move slow. Bless you. So you manage to separate proteins based on the size. The gel, though, is awfully inconvenient to work with. Imagine that you have to work with quite literally, well, not literally, but you have a jello as the experimental substance. It's not great. So you want to transfer those proteins, that separation pattern, onto some medium that is easier to work with. During blotting, right here, you apply electricity so that proteins will go from the gel onto a special material called a membrane. It's like a paper. Okay, make sense? You making a copy, like you basically making a Xerox copy of your gel on the membrane. Am I making sense right now? Remember, those proteins are separated. You know which antigen you're looking for. You know its size, you know where it's supposed to be. So you add antibodies, and if your antigen is present where you expect it to be, antibodies will bind to it and you will see a signal. Does that make sense? If it's not present, no signal. Well, I like this method. It's cool. You feel like scientist when you do that. The problem is it takes a lot of time. It has low processivity, so you can analyze a lot of samples. It is not quantitative. Very sensitive, though. But the problem is, let's say I'm looking for Crimean Congo hemorrhagic fever antigen. If I don't find it, it doesn't mean that the patient's healthy. It means that they don't have Crimean Congo hemorrhagic fever. There's something else. That makes sense? So, in an attempt to fix, quote unquote, the problem with processivity, we have a method called ELISA. I'm going to focus on the direct assay. You don't need to distinguish between direct sandwich and indirect assay. But the idea is this. You take your sample and you put it in the well of a plastic plate, okay? And your antigen, well, actually, all proteins get stuck to the bottom of the plate. We're not going to get into chemistry of this, but it can be done, okay? Make sense? So they get stuck. You're with me, right? You wash your samples, you add antibodies. These antibodies have some kind of an enzyme, some kind of something that will produce a signal. So if the antigen is in the well, antibodies are going to get stuck, you will see a signal. If antigen is not in the well, antibodies aren't going to get stuck, you will see no signal. Am I making sense? Obviously, the more antigen you have, the greater signal you're going to get. Okay? So the advantage of ELISA is an acronym, by the way, it has very high processivity. You can automate it, and you can make it quantitative. The problem with what if we don't find anything, what's going on, still remains. Finally, the third method that I wanted to mention fairly briefly is immunofluorescence. So you see this fork with 4 green dots on its handle. Can you see that? So that fork is an antibody. Those green dots are special molecules that can fluoresce under the black light. So what do we use it for? Let's say you study some patient who succumbed to a very rare viral disease, and you want to know where the virus goes into in the human body. Am I making sense? So you say, I want to see if the virus is in the kidneys. So remember, the person's dead already. So take kidneys, make a bunch of slices of those kidneys, and you apply the antibodies to that extremely rare virus. And if it's present, antibodies are going to stick to it, and you're going to see fluorescence. If there's fluorescence, there's virus. It's not a quantitative method, but it is really useful to identify which organs are infected. Does that make sense? So the questions that you can expect overall about this methods will be focused on matching the term for the method with what's going on. Are we good? Let's move on. Biochemical methods. This is one of my favorite methods. It's super cool. I know that the image and the term, they are super intimidating. My goal is to break it down for you. So I want you to completely ignore right now the text on the slide. I want you to focus on the image that I have in the middle of the screen. So here's what's going on in this method. I don't name it yet. First, you have your bacterial sample, and you mix your bacteria with a matrix. This is highlighted as the mixture of bacteria with the matrix. Clear, right? Then you use a laser to blast the living shit out of that mixture of matrix and bacteria. Okay? So now let's go to the name. You mix bacteria with matrix, therefore matrix assisted. And then you blast your sample with a laser. So laser desorption. Do you follow? When you blast this sample, you not just evaporate, you break down the cells into a bunch of individual fragments. That's ionization. These fragments, you can see them right here. These are the fragments. They have different mass, and they have different charge. Keep up with me for a minute. And then you apply electromagnetic field to those fragments, and because they charged, they start flying through that field. So question number 1, which particles will fly faster? The one with a larger or smaller charge? Larger charge. Larger charge will make them fly faster. Clear? Which means the time of flight is going to be shorter. Time of flight. Clear? What about mass? Which particles will fly faster, small or large? Small. So small particles will have smaller time of flight. So you effectively have two parameters that determine the time of flight. It's the mass and the charge. The smaller the mass, the shorter the time of flight. The smaller the charge, the longer the time of flight. So you effectively categorize your particles based on the mass to charge ratio. Does it make sense to you all? So these particles will fly and they will hit the detector, and detector will record an image like that with peaks. Now remember, those peaks, they represent the fragments of cells. I have another question for you. So let's say you go to IKEA and you buy a set of 6 cups. Identical cups. Same box, everything. You put all the cups in the same height and you drop it on the floor into the same spot, one after another. Will they break in the same pieces or not? No. Well, thankfully bacteria are not IKEA cups. If you blast them with the same intensity of a laser beam, they mixed with the same matrix at the same concentration, you reproduce all the conditions, they're going to be exploded into the same fragments. So what it means is that you can take a known bacterial species and do this MALDI-TOF spectrometry several times and produce a unique signature seen on the left. And when you have a patient coming in and you collected bacteria and produced pure culture and, ran it through spectrometer, and the image matches your database entry, you identified your bacteria. Isn't that cool? Like it works, okay? So the plus side, you load your sample, you click the button, 2 hours later you get the result. If it is in the database, you're going to know what it is. Downsides, you need pure culture. Takes time. Well, if it's not in a database, you're not getting the result. But when I learned about that machine being introduced, which was before pandemic, back then, the price tag on one was $750. Sorry, not hundreds. So only big hospitals can afford it. Does that make sense? So you can hardly expect something like that in like a local hospital in the middle of nowhere. Sorry. Finally, genetic methods. So there are two that I want to highlight here. I believe you've heard the name of one of them. PCR, polymerase chain reaction. So here's the thing. There's an enzyme that can multiply DNA molecules called DNA polymerase. Okay, all good? Different organisms have unique DNAs. Okay? What you can do, you can design short DNA fragments called primers that will bind only to the DNA of a very specific organism. You can design primers that will bind only to human gene for, I don't know, apolipoprotein 4E, whatever, okay? Only to that gene. You can design primers that will bind to the operator of lac operon in E. coli. You can design primers that will bind only to the gene, the spike gene of coronavirus. The point is, these primers are going to bind only to those fragments, only to that DNA fragment and nothing else. Clear? You add primers that bound a certain DNA fragment, and you add the enzyme. And lo and behold, this space between the primers, this DNA between the primers, start getting doubled. One cycle, it's doubled. 2 cycles, it's quadrupled. 3 cycles, 8 times more. 16, and you just go exponentially. After 40 cycles, increase the number of that specific fragment by about 1 trillion times. Slightly more than that. So, when you have that one little fragment of a known size that is so much amplified, you can detect it. Am I making sense? Right? You can detect it. I don't want to get into details how, but you can. If it is there, your DNA from a microorganism is there. Doesn't necessarily mean that actual live organism is there. I'll get back to it in a second, but so far, am I making sense? The advantage, super sensitive, can be automated, can be very easily made quantifiable. Okay? Disadvantage, if your primers are wrong, you're not getting any result. That's it. That make sense? And sometimes being too sensitive becomes a curse. There were multiple examples during COVID pandemic when after PCR test, people were considered positive. You know, the test came positive. And when test was redone, it was still positive, okay? But the problem is, PCR is such a sensitive method that those people, some of the people, some very small amount, some people who were positive effectively did not have any virus in them. They just had fragments of the viral genetic, viral genome. Does that make sense? Not actual virus. Go ahead.
So, if it's that sensitive, like, if you were running 2 tests side by side, like the wind blew, couldn't you contaminate it and then also get
Absolutely. Doing PCR, avoiding contamination is a massive pain in the butt. 100%. I can tell you this, if you work on SARS-CoV-2 and I work with bacteria and I isolate bacterial DNA and we work in the same room, different biosafety hoods, different places, just same room, your samples are going to be 100% contaminated by my bacterial DNA. DNA is a problem, it's everywhere. Not my words, I'm quoting a scientist. Okay, yes, it's a huge problem. This is why primer design has to be so impeccable. Because if your primers, your red fragments can only bind to unique sequences in the coronavirus, my bacterial DNA is not an issue. Like, it's there, but it's not detected. But technically, yes. If your primers can pick up something similar, you bound. Okay? That makes sense? Now, why it is important to distinguish between the detection of genetic material and the detection of actual microorganisms. Absolutely real. I'm proud of it. I'm giving you very much updated information of infectious diseases. So there are several outbreaks of Legionnaire's disease in the United States. Are you familiar? You're familiar with Legionnaire's disease? It's a bacterial infection. It's a typical pneumonia. It cannot be transmitted from person to person. People get it when they inhale water droplets. Okay? Do follow. So when people inhale them, they get pneumonia. Now it's treatable. Before it was pretty bad. Mostly the source are cooling towers with the water surface on top. Okay? So during the outbreak, like last 3 weeks, there's a bunch of cases, I believe mostly in New England. So local departments of health went through the towers and tested them by PCR for the presence of genetic material of Legionella. Okay? Let's say, I don't remember, let's say they found genetic material in 15 towers. They surveyed these 15 towers for actual bacteria. Found it in like 5, which means DNA may be just the fragment of a cell. It doesn't have to be the part of a live cell. Am I making sense? So distinguish those things. Finally, sequencing. So when I was in college, we studied chemical and enzymatic sequencing of DNA, and it made sense. I understand how it works. Then, many years later, next generation sequencing appeared, and I was curious, and I went into technical details, and I realized that people who came up with the next generation sequencing, no wonder most of them are from California, because you have to smoke a very heavy weed to come up with those ideas. Like, this is insane. What they proposed, the technology is absolutely crazy. So what is sequencing? You determine the sequence of all DNA or RNA in your sample, which is a blessing and a curse. Let's say I have a patient with some, unusual disease, right? This is what happened in the beginning of the COVID pandemic in China. They had patients with typical pneumonia, they collected samples, and they sequenced all DNA and all RNA. So they get everything from human cells, from bacteria, from everything that person has. And you have to dig into this pile of shit and find an actual signal, find what is unusual. Obviously, humans don't do it, software does it. But again, what if you find 2 or 3 unusual sequences? Which one is that? Am I making sense? It's super sensitive, but it provides so much information that sometimes it can be daunting. So to conclude this, regarding the identification techniques, you're going to see which of the following statements is correct about Western blot. Protein samples is separated. A protein sample is separated in the gel, transferred to a membrane, and the presence of antigen is detected by antibodies. There you go. Does that make sense? The term for the method and its description. Questions? All right, folks. Let's wrap it up.